Hello, everyone. This week we're going to be talking about, along with human thermoregulation, skin tone, as well as high altitude adaptations. We're also going to talk about blood, right? And why do we have different blood groups in humans, and why do we have different blood types, right? And I threw this graphic up here because it's kind of humorous, right? But we're going to talk about something that's a bit interesting regarding mosquitoes and our blood type. And we're going to find out that in a lot of cases, our variations in blood type that we have on a very basic level are generally an evolutionary response to some sort of disease pressure that is has killed humans in the past, right? So, and we'll talk about that. Each of our different blood types can kind of be considered a polymorphism. It's a slightly different variation of the same kind of chromosome that governs the production of blood. So we're going to take a look at all these different factors. We're going to look at what really constitutes blood, what its different components are, and we're also going to take a look at a little bit of history and some fun facts about blood as well. So the central questions we'll be dealing with is what type of diversity do we see in human blood groups? Why do humans have different blood groups? What determines how many blood groups are within a given species, right? And how do other cultures view blood? So we'll talk a little bit about the functions and properties of blood, things like plasma versus blood cell production. We'll talk about erythrocytes as well as blood types, leukocytes, as well as hemostasis. So looking at a little bit of blood and history before we get into the nitty-gritty details about what exactly is blood. Some of the first writings that we have that reference blood directly in China at around 1000 BC, we have writings that say that the soul was contained within the blood, right? We also have instances or recordings of Egyptians that bathed in blood for their health purposes, mainly those of the upper class and pharaohs and such. We also have Pliny and Celsus from who describe Romans drinking the blood of fallen gladiators to gain strength and vitality and to, of course, cure epilepsy as well. We also have taurobolium, which is the practice of bathing in blood as it cascaded from a sacrificial bull, which was practiced by the Romans as well. So blood has always played kind of an integral role both biologically and culturally for our species. Moving a bit further in time, our first blood transfusion was in 1667 by Jean-Baptiste Denis. Transfuses blood, lamb's blood, into a French convict. Right. As a matter of fact, all of these early kind of transfusions were done on these kind of convicts and people who belonged to homes for the mentally ill. And it's really kind of interesting because most countries had laws against transfusion until the 19th century because a lot of these early experiments used all sorts of different materials for transfusing. Remember, this is before they had rubber tubing, which is what we use now for transfusing. So in essence, they were using things like intestines in order to transfuse blood from one individual to another. They were also transfusing all sorts of nasty chemicals or all sorts of different materials like milk, orange juice, even pig semen was used and transfused into someone to see what the effects would be. So naturally, most of these people who were in these early transfusions died, right? And even the ones that were transfused with actual human blood, that was before we knew about things like agglutination and how your blood can clot if you don't get the appropriate blood type being transfused into you. So most of the early experiments were failures, right, which led to these legal prohibitions against transfusing. So to look at some general patterns that we see in kind of cultural responses to blood, drinking blood on an average, if we look at all human cultures across the globe, there's generally a strong taboo in most cultures against drinking human blood, except in cultures that use blood sausage and blood pudding, both of which are traditional dishes in European cultures. They don't use human blood, but these are dishes that are made specifically out of animals' blood. So the question really is somewhere in our past, was there an evolutionary undercurrent to the development of these taboos to prevent the transmission of disease. We also have a strong taboo in most cultures against menstrual blood when women are menstruating. In many cultures, particularly those in New Guinea, the tribal cultures like the Dani, they're seen as kind of polluting or corrupting. So when women are in menstruation, they will use these menstrual huts and sequester themselves until they stop bleeding, right? Because there's a cultural taboo against coming into contact with menstrual blood. If we look at another archaeological reference to blood, one that I'm more familiar with because I've have experience working with Mayan hieroglyphic epigraphy, there are bloodletting rituals which were very common amongst the Maya elite. Where actually, if you look at the photo featured to the right here, you see one going on. It's used as a way to communicate with the ancestors because in the Maya religion, they believe that blood quote unquote feeds the gods. So bloodletting rituals also differed based on the sex. For men, the men would take a 12-inch stingray spine from a stingray and insert it and pierce their own genitals and then collect the blood and burn the blood. And they believed in their religion that the spirits of their ancestors would come forth out of the smoke of their own burning blood. For women, on the other hand, what they would do is they would take a rope that has been that has certain barbs placed on it, and they would create a hole in their tongue and run the rope through their tongue. That's actually what you're seeing featured in the image here. And the ancient Maya, these people lived in Mexico, Belize, and Guatemala. This was actually a relatively common ritual and ceremony that was done amongst kings and queens, right? This actually wasn't something that was done amongst the common person. So moving on to some more of the kind of modern kind of hypotheses and kind of folk theories, quote unquote, or folk hypotheses that run around blood today. We have this notion of the modern Japanese blood type personality theory, is what they call it. It's basically a folk science hypothesis from Japan that personality traits can be directly linked to blood types. And just so you know, this has no actual basis in science, and it's similar to something we're going to talk about here in a minute called blood type dietary theories, which lack any actual valid data supporting their existence. So according to kind of this folk hypothesis from Japan, and they get really complicated. I'm not going to go over every single personality trait, but you'll see in the next couple slides that they can get pretty complex with what they believe comes through with certain blood types, right? So, but in a general sense, they say if you're a type A blood, you should have your personality is of a well organized person. If you have type B blood, you are selfish. If you have type O blood, you're optimistic. If you have type AB blood, you are quote unquote eccentric, right? I guess getting the mix between well organized and selfish. So you can kind of see how this would be a bit deterministic. You know, we talked about early in the semester biological determinism, right? This is a biologically deterministic hypothesis, right? Basically saying that your biology dictates your personality, and that your environment and your rearing and all of those things had no part in the personality. It's all related to what blood type you have. And you can also see how this would also make it seem like, you know, everyone of type A blood should kind of have the same personality, which we all know is most likely not true, right? So you can kind of see how this is, on its face value, sounds kind of interesting, but really no way to actually scientifically prove this or really give it much weight. There's also another kind of interesting modern hypothesis trend that we see in this kind of folk science regarding blood types. We have the blood types and diet, and it was created initially by a guy named Peter D'Adamo, who is a self-proclaimed naturopath, which is not a real profession. There is a specific ideal diet, essentially, for each blood type. This is what Peter Adamo or D'Adamo claims. And he says you can prevent diseases like cancer, diabetes, following the correct diet for your blood type. And we know this to be a little bit ridiculous because diabetes is a complex disorder that is caused both by a genetic factor as well as an environmental factor. And cancers, we talked about germline mutations, right? These are cause of a huge degree of cancer or causes of cancer, and you also have environmental causes of cancer. Yes, your diet does play a role in terms of your DNA's ability to repair itself and your overall sense of health, right? But diet does not prevent disease quite like Peter D'Adamo and these proponents of the blood type diet hypothesis really are kind of claiming. So to just give you an example, the next couple of slides are going to kind of show you what's going to happen or what this breakdown is of kind of how they believe diet affects blood type. I'm not going to go over each specific blood type and what diet you're supposed to have because we all know it's really junk science. Because we know that humans as a basic concept are generalized species, right? And unless you have a genetic condition that leads to a specific dietary condition, in most cases, humans in a general sense can really, at different age brackets, eat the same relative diet and maintain healthy lives. So let's look at some of the basic functions and properties of blood, right? People on average have between 4 to 6 liters of blood in them at any given time. And there are two basic components, which include the clear fluid, which is known as plasma, and then you have the cells and the platelets, which include your erythrocytes, your red blood cells, and your leukocytes, which are your white blood cells, right? And when you do the process of centrifuging, or when scientists centrifuge blood in a lab, which is the process of placing it in a machine and subjecting it to very, very high degrees of rotation, those two parts will actually separate. You'll actually get the plasma, which comes off at the top, followed by a layer of white blood cells, and the red blood cells, being heavier because they contain a lot of iron, are going to sink to the bottom. So red blood cells themselves make up about 45% of the volume, right? A number that we call the hematocrit, right? So you can have disorders and genetic conditions where you have higher than normal hematocrit levels within your body, or you could have a dietary deficiency where you have causes lower than normal hematocrit levels. Red blood cells make blood 4 times as viscous as water, right? That's why blood is much thicker than water. And we talked about when you go up into high altitude environments, you begin to produce more red blood cells, right? So you can imagine how viscous and how thick your blood can become if you're not really accustomed or you don't acclimatize properly to that high altitude environment. So looking at our basic blood components, right, plasma is about 55% of the total blood volume. Plasma itself is 91% water, about 7% blood proteins, which like albumin and globulin, 2% nutrients like amino acids, sugars, and lipids. There's some hormones in there as well as electrolytes. The cellular components of blood, to break them down, you have, remember, it's about 45% of total blood volume. You have the buffy coat, which is the white blood cells, and the platelets, as well as the heavier red blood cells. To go into a little more detail about your erythrocytes or your red blood cells, they are both an oxygen and a CO2 carrier, right? They exchange those two molecules. They determine your blood type. They need to be resilient in order to get through your capillaries. That's why red blood cells and the protein, the hemoglobin molecule, is shaped the way it is, right? It's circular so that it actually has a certain degree of aerodynamic capability to it. Hemoglobins make up about 33% of the cytoplasm. The nucleus is lost during cell formation when we look at these red blood cells. So overall, your red blood cell count or your hematocrit levels will tell how much O2 your blood will be carrying. And women tend to actually have lower hematocrit levels than men do on average. And we think that this is because androgens stimulate red blood cell production. We also think this could be due to the monthly menstrual loss as well. And it could be inverse proportion to body fat, right? So and males also, on average, clot faster. So what evolutionary significance might this have, right? So the question really is we do see very, very minor differences in terms of between men and women in the human species overall, but in terms of these differences across quote unquote races or any type of human populations, they don't really exist, right? So we have slight differences between men and women, but not across other constituent categories that we've created. So looking at one of the polymorphisms relating to the gene that produces your blood cells, we have chromosome 11, which carries the code for the beta chain of that hemoglobin molecule. And the normal allele there is called hemoglobin A, right, or HbA for short. The sickle cell allele is called hemoglobin S, right, which produces an abnormal hemoglobin, right? This is the polymorphism that is at this area, right? Now, I know what you're thinking, when we generally see the word sickle cell together, it relates to sickle cell disease, which is a deleterious disease that generally kills people without having regular medical care. But we're going to talk about a situation of why did this disease pop up in the first place and what response humans have made to certain selective pressures that have caused that mutation to occur. So if we look in a general sense, right, why do we have polymorphisms at this ABO blood locus on chromosome 11? Well, this is due to these different disease pressures, these different selective pressures that our species has had to deal with throughout our evolutionary history on the planet, right? If we look at the studies, they say that the B allele helps protect against infant diarrhea, which is a primary cause of death for infants in places that don't have modern medical treatment. We have A and B alleles help protect against the plague if you have them both together. The O allele helps protect against bronchial pneumonia. And if we go even further, we can say different specific Rhesus groups like the Rh plus allele we find is more susceptible to smallpox. And that actually has a very interesting and a sad story behind it. Essentially, we know that most Native Americans belong to the four same haplogroups, right? It's called A, B, C, and D. And what this indicates is that Native Americans originally came from four separate and distinct genetic stocks that all came from that Asiatic region. And what ended up happening is each one of those stocks had a high degree of Rh plus alleles within their population. So most Native Americans at the time of European contact had the Rh plus allele in their blood system, which made them more susceptible to smallpox. This is one of the reasons why smallpox was so virulent and killed so many Native Americans really before the Europeans even colonized North America. So if we look at the B-type blood, it offers some resistance to malaria. Mothers can pass innate immunity to infant diarrhea within their breast milk. And this particular blood type originated roughly on our planet around 3.5 million years ago. So this is the result of the selective pressure to a specific disease, right? So this didn't just affect us, it actually affected one of our ancient ancestors, most likely either Ardipithecus ramidus or Australopithecine afarensis. So it's actually really interesting that we're seeing a blood type differentiation that occurred within our evolutionary lineage that has lasted the test of time because it has provided us with a slight advantage to this very serious disease. The components of type A blood are actually extraordinarily old. We see them in other mammalian species, and we know that the earliest recorded instance of type A blood types within mammalian species occurred before humans between roughly 9 to 11 million years ago. And we know that it protects slightly against the plague, but it's very susceptible to complications from malarial infections, right? As a matter of fact, people with type A blood tend to be able to carry far more of the malarial parasites than other blood types. So if we look at type O blood, it protects against bronchial pneumonia, offers some resistance to the malarial parasite, and is found most commonly in Africa and Europe. And we're still kind of out on how old this actual blood type is. It's the universal blood type donor, so we hypothesize that it's an exceptionally old blood system within the mammalian DNA. So going back to our example that we're going to use of demonstrating how human blood type systems have evolved to respond to diseases, we go back to the HBS allele. Remember, that's on chromosome 11. And what ends up happening here is it causes a substitution of a thymine with a uracil. And if you have normal blood cell coding, it essentially goes from a thymine to an adenine, and it produces a macromolecule called glutamate. If you have the abnormal allele there, the HBS allele or the HBS blood cell, you have adenine that goes to uracil, which produces valine as a replacement amino acid for that glutamate. So in terms of human populations today, what we generally see in terms of this HBS allele and its distribution is that if you have both of the kind of normal copies, of course termed homozygous, that we generally find European and Asian populations that have this in the highest degree. In terms of the HBS allele having one normal allele and one affected allele being malarial resistant, we see this in some West Africans as well as Central African populations. We also see having both copies of the affected allele, thus resulting in sickle cell anemia, which occurs in some West African populations and Central African populations as well. So we also, just to kind of talk about how this relates to human evolution in general, the HBS allele is not the only one. We actually have 4 identified different sickle cell variants among different African populations, suggesting that the same mutation arose independently among the populations at several different times, right? We have the HBV gene, we have the HBC and the HBE alleles, which arose and spread across Africa as well as in Southeast Asia. So why is this all important, and why are we talking about the HBS allele and the polymorphisms on chromosome 11? Well, it's because malaria is a parasite that attaches to your blood system, and we'll talk about, or the video will show kind of what is the result if you get infected with malaria. Generally, you get fever, chills. It's a very kind of serious illness that in most cases results in death. If you have one copy of that affected allele, what it does is it makes your red blood cells slightly misshapen, not as quite as bad as the sickle cell that you see featured here, because this would be if you had 2 copies of the allele, but it makes it just slightly sickle-shaped, which actually prevents the malaria parasite, or most of them, from being able to attach to that red blood cell. So in essence, you short-circuit the malarial life cycle. It won't prevent you from becoming infected, but it will. You have some natural resistance to the more deleterious side of the malarial infection. Now, if you have both copies of the normal allele, your blood cells are normal shaped, and malaria is going to ravage your body, and you will most likely die. On the other hand, if you inherit both copies of the allele, the sickle cell allele, you get sickle cell anemia. That means that your cells are very misshapen, and you actually have trouble transporting oxygen to tissues, and have all sorts of systemic problems as a result. So if we look at kind of the distribution of both the HBS allele as well as malaria, you're going to see that the maps match up nearly perfectly, right? We see the HBS allele in all these areas in which have very, very high presence of malaria, right? So in essence, what we're seeing in live time, or what we see has happened in our past, is this kind of evolutionary response to a very significant disease pressure. Now, although malaria or levels of malarial infections are very low today, since this is a genetic condition or genetic change that's occurred in our DNA that does not really affect you before reproduction, in essence, that's why it continues to be passed on, and it does have an advantageous effect still in areas in which there's high degrees of malarial infections. So in essence, it's a very bad trade-off kind of for type A blood types who already have larger than normal blood cells. What ends up happening is in the for type A, if your blood cells are slightly misshapen into a slight sickle shape, your blood may actually start to get caught up at the capillary junctions in your capillary system, in your arterial system, and you'll end up being more susceptible to things like embolisms, things like heart attacks and strokes, and all those kind of cardiovascular events. So to give a few details about sickle cell anemia, it's a hereditary anemia that's found in African American populations as well as African populations. It causes an abnormal sickle shape of the erythrocyte or your red blood cell. It's thought to have developed as a defense against malaria, which we pretty much know at this point. The symptoms can be very mild to very severe, right? You can have very mild, like pain in the back of the legs or in the abdomen, which is most common, some fatigue, a little bit of irritability, but it can also get really bad to the point where you have chronically swollen joints, you have open leg sores, as well as gum disease. So you also have a something called a sickle cell crisis, which is a group of symptoms, and the pain is kind of compared to that of cancer pain. We also see another polymorphism on that chromosome 11 called G6PD deficiency. We also kind of more commonly kind of call it as favism because with this particular deficiency, you can't not eat fava beans, or it could be very damaging to your red blood cell system as well as your kidneys. But essentially, we find this G6PD deficiency in a lot of Mediterranean populations, and when we look at their response to malaria, G6PD deficiency also allows them to be slightly resistant to malaria as well, but they have to be careful when reproducing. They also have to be careful when taking certain oxidative drugs like aspirin or sulfonamides. They'll also have to be kind of careful when they're eating different foods, right? Like because they can't eat fava beans. So what happens in G6PD deficiency if you don't follow the dietary restrictions and you don't follow the drug restrictions? We know that it itself is an X-linked trait, so that's why it's hereditary. It causes an oxidation of the sulfohydryl proteins within your cell, and that oxidation causes protein denaturation, which basically just kills the protein and makes it unusable, which results in insoluble cellular detritus called Heinz bodies. This is garbage, essentially, that your cells can't deal with and can't get rid of. So what this results in is hemolytic anemia, where you have a bunch of basically dead red blood cells floating around your body, and you're not able to produce and recycle enough of those red blood cells to keep up with the destruction that's occurring as a result of the G6PD deficiency. So this, much like the HBS allele is a hereditary condition, so you can kind of see kind of how it moves through a population or moves from generation to generation. This is what we call kind of a pedigree chart, which shows how this gene or how this condition has moved and how it will affect the next generation if the given couple have 4 children. We also have another polymorphism that results in a condition called thalassemia, which results in a malformed red blood cell. And this is another kind of condition that we see in high prevalence in areas where the malaria infections are very high, right? So this is just another polymorphism. Yes, it does result in deleterious effects later on in life. You may you have a higher risk of death from heart attack, you have a higher risk of stroke. But the trade-off is that you live long enough to reproduce, right? So you stave off that malarial infection or the deleterious effects of it for long enough to be able to reproduce, which, of course, is the name of the game in evolution, right? So in terms of your thalassemia, the condition varies depending on what you have inherited. If you have severe thalassemia, also called alpha thalassemia, you will have abnormal metabolic function and you'll have lower than normal blood cell count. If you have mild to thalassemia, it's referred to as beta thalassemia, you'll just have abnormal levels of blood cells, right? Severity depends on how many factors are inherited from the parents or lack thereof, right? So what we've been talking about here with our G6PD deficiency, our HBS allele, our variant that causes thalassemia, what we're really talking about here is a balanced polymorphism, right? This is where the heterozygote individual has the greatest fitness and the greatest reproductive success within those given populations, right? If you have both the normal alleles, you're going to catch malaria and die. If you have both of the affected alleles, you're going to get sickle cell anemia and die, right? So you want to have one of the affected alleles and one normal allele, so you are the heterozygote, so that you have that greatest reproductive ability, right? But you must be careful when reproducing because, of course, if you find another person who is a heterozygote, 2 out of the 4 children are going to be negatively affected by their kind of genetic makeup. If we look at our RH plus groups, or otherwise known as our Rhesus groups, there are 3 genes that control these groups. You have C, D, and E, and each with 2 alleles. So you can have DD, or we know all the general genotypes you can formulate with those. And those are what produce the antigens on your red blood cells, right? They're classified as either RH plus or Rh negative if you lack those D antigens. So combined with the ABO blood group to get blood types like A positive or B negative, right? So that positive and negative in your blood type is referring to what amount of these D antigens you have or whether you have them or not. Along with the general transfusion problems you can get if you have the wrong blood type in general, you can also have problems if you're transfused with the wrong Rhesus group, right? If you're an Rh negative person and you receive Rh plus blood, the first time that transfusion happens, you may be okay. The body hasn't made any anti-D antibodies yet, but the second transfusion, if you receive a incorrect transfusion again, it can cause severe problems. With fetuses that have different Rh groups, the pregnancy is fine as long as there is no tearing of the placenta and causing a mixing of the 2 blood types, right? Then the baby might be born with a disease called hemolytic disease of the newborn, which is a type of blood anemia. So what happens if you receive the wrong blood type in general? You have this process called agglutination, right, which will cause your red blood cells to block your arterial system and cause those vessels to rupture. And this will cause some free HB in your system, which can block kidney tubules and cause death. So universal donors and recipients. So a universal AB is called our universal recipient because it lacks both antibodies for A and B, and O is called our universal donor. The problem is the donor's plasma may have antibodies against the recipient's red blood cells, right? This is why we do extensive testing when we go to do blood transfusions, right? You want to make sure that the blood type is correct as well as the Rhesus group being correct. So if we look at our other blood groups, this is really where we see the vast degree of variation within the human population. We have over 100 other different blood groups as well as 500 different types of antigens. You have the Duffy group, the Kell group, the Kidd group, and the Lewis groups. It goes on and on and on and on. They rarely cause transfusion problems, and they're useful in paternity tests. So, and they are also useful in showing how human migration patterns have occurred in the past, right? So you can actually trace these different Rhesus groups and find migration patterns. So your blood system overall is kind of a very intricate system that requires a certain degree of balance between your leukocytes and your erythrocytes, right? So looking at kind of what happens if you have too much or too little of one, looking at your red blood cells, if you have a too high volume, you'll have reddened skin tone, bloodshot eyes, you'll have increased blood volume and pressure, and an increased workload on your heart, right? This is what high blood pressure is. If you have too low of volume, this will lead to anemia, headaches, fatigue, pallor. It can even lead to things like shortness of breath and fainting. If we look at white blood cells or your leukocytes, if you have too many or too high of a volume, you'll have too acute of a response to infections. If you have too low of a volume, you have a weakened immune system and be more prone to infections. If you have a high degree of too many platelets, you'll get thrombocytosis, which there's really no serious side effects. If you have too little of a volume, you'll get clotting problems, bruising, nosebleeds, as well as blood lesions. So let's look at an example of the intersection between biology, which would be our different blood systems, and culture, right, which we're going to talk about marriage practices and European royalty and hemophilia. So hemophilia itself is an X-linked trait, but some get it as a spontaneous mutation. It essentially causes a trouble with clotting factor 8, and the incidence of hemophilia is about 1 in 7,500 live male births and about 1 in 25 million live female births, right? It's low because we can ID it in modern populations and address it accordingly, right? And it has a very, very high prevalence amongst males versus females, right? So to give you some more details, hemophilia is the genetic lack of any clotting factor that affects coagulation. It's a sex-linked recessive trait in males. It's inherited from your mother. Hemophilia A, and there's a few different classifications, is missing factor 8. It's about 83% of the cases. Hemophilia B is missing factor 9, which is about 15% of the cases. Hemophilia C is missing factor 11, which is autosomal. So this is one that would be the result of a spontaneous mutation. And the disease, we know that a physical exertion causes bleeding and excruciating pain, which ends up leading to swollen joints as well as lesions on the skin. If we look at the earliest written recording, there's a recognition of hemophilia is in the 2nd century AD in some Middle Eastern Jewish writings. Females are usually carriers since they have a chromosomal makeup of 2 X's, right? Inheriting an X chromosome with disease mutation will generally not produce a disease condition as the other normal X chromosome can compensate, right? So hemophilia affects males much more frequently than females. We talked about that with the live birth statistics. And the spontaneous mutation can occur in 33% of patients. There really is no family history of hemophilia today. So it's really interesting that we've recognized this kind of disease within kind of our genetic makeup. We've kind of made it aware. Some people have, still decided to have children. Some people haven't. We've been able to get genetic testing and everything like that. So in terms of the inherited factor of hemophilia, those occurrences have gone down, right? But we still kind of see that spontaneous mutation hemophilia, that autosomal condition. So hemophilia became known as the royal disease because it was spread through the royal houses of Europe through the marriage practices and through the marriage customs and through the marrying of a lot of Queen Victoria of England's granddaughters and grandsons to various royalty across Europe. So if we look at one specific famous example, we have Alix of Hesse was one of Victoria's granddaughters and married to Tsar Nicholas II of Russia, becoming Tsarina Alexandra. She was a carrier, and her fifth child and only son, the actual heir, Alexei, who was born in 1904, was a hemophiliac, right? And Alexei's hemophilia was discovered shortly after birth because of excessive bleeding from his umbilical cord. And the kind of boy led a pretty rough life with a lot of doctor's visits because of swollen joints. He dealt with excessive bleeding. He had to be exceptionally careful of even getting very minor cuts because he lacked that clotting factor to allow his wounds to scab over and prevent further blood loss. We also have hemophilia popping up through Victoria's granddaughter Irene of Hesse, the Princess of Prussia. She had three sons, two of whom were listed as hemophiliacs. If we look at another example, we have the Princess Victoria Eugenie of Battenberg. She was another one of Victoria's granddaughters who was married to King Alfonso XIII of Spain, and she gave birth to 7 children, 2 of which were hemophiliac sons and only 1 healthy son, right? The rest were healthy girls. So why is this all important, right, in terms of human cultures and human biology and evolution, right? Well, the spread of hemophilia in the royal houses of Europe is the result of strict cultural marriage practices, right? So, we may hypothesize that, marriage practices may have originally developed as a way to build alliances as well as prevent the spread of disease. Unfortunately, they didn't really know about certain genetic diseases, right? This is before genes and gene theory was around. So hemophilia is more common today than it was 100 years ago. So to give you kind of the other end of the spectrum, we also have coagulation disorders or unwanted coagulation, right? With our hemophilia, that's a disease in which you have unwanted bleeding. In these conditions, you have unwanted clotting or unwanted coagulation. We have embolisms, which is an unwanted clot traveling in a vessel. You also have thrombosis, which is an abnormal clot in an unbroken vessel. This is most likely to occur in leg veins of people who are inactive, right? So you should walk at least about 30 minutes a day. The clot will travel from veins to the lungs, producing a pulmonary embolism, and death from hypoxia will occur. I'm not going to describe the process, but just to give you a brief summary, it is basically drowning on your own blood. So infarction or tissue death may occur if the clot ends up blocking a blood supply to a major organ. It could cause a myocardial infarction or a stroke. Other blood disorders include anemia, which is a shortness of red blood cells. Of course, we talked about hemophilia. We have thrombocythemia, which is an abnormal small number of platelets in the circulating blood. We have hemochromatosis, which is a disorder of iron metabolism characterized by excessive absorption. You have Hodgkin's disease, marked by chronic enlargement of the lymph nodes. You have leukemias, which is progressive proliferation of abnormal leukocytes. You also have non-Hodgkin's lymphoma, which is lymphoma other than Hodgkin's disease, essentially. So you have basically enlargement in other areas. So in terms of evolution, why did we differentiate in blood types, right? It's easy to kind of say, in a general sense, it's due to the pressures or selective pressures the disease placed on us, but we also have two competing theories that really talk about it in more detail. We have the variation in selection, that different types are selected for at different points in history by disease pressures. So some people with type A blood will survive at a certain period during a certain pandemic, and they'll increase those numbers in the gene pool, and then later on they'll become more effective. That's kind of the pattern of variation in natural selection. Or we have negative frequency dependent selection, which is rare blood types have more advantage, right? Bacteria adapt to the most common host, right? This is the same with viruses as well as parasites. So having a differentiation or having a more rare blood type, in theory, makes infection rates low, right? So you may actually, they hypothesize that it's really these unique blood types which are more evolved than kind of our more basic blood types. So to wrap everything up, humans have different blood types as a response to environmental selective pressures at different points in our evolutionary history, right? Each blood type has an advantage and disadvantage in terms of immunity to certain diseases. People with certain blood variants, particularly those that are like the HBS allele and things like that, must be careful when reproducing. Without diversity in blood types, humans would be more susceptible to extinction via disease.